Namaste everyone. We are going to demonstrate negative geotaxis assay in Drosophila melanogaster. This experiment is widely used to study locomotor defect and neurodegeneration. This experiment is performed following the standard procedure described in chapter 92 of this reference book named Experiments with Drosophila for Biology Courses. The link to this book is provided in the description box below this video. For this experiment, required materials are climbing assay apparatus, funnel, foam, ruler, marker, vials containing flies of two different days in which we use Oregon R plus as a control and Park 1 mutant to compare locomotor defect. Before starting the experiment, a ring should be marked on a test tube using marker and ruler at 10 cm height from the bottom. So the experiment starts with gently transferring the flies of different strain to different test tube. Make sure while transferring the flies into the test tube, gently tap it on a foam so that it do not break during tapping. At last, the flies are tapped gently to bring them to the bottom of the test tube and observe their movement for a fixed duration, usually for a 10 seconds. Record the number of flies that cross the mark height within the given time. To calculate percentage climbing activity, given formula is used. So this is our data which we have recorded during this experiment. It can be easily seen that percentage climbing activity of Oregon R plus does not show much variation at day 3, 7 and day 10. On the other hand, the Park 1 mutant show progressive decline in percentage climbing activity as it decreased from 40% to 30% and then to 20% if we see from day 3 to day 10. One thing has to be noted here that this is one set of our experimental data. You can perform this experiment in multiple sets to obtain statistically significant result. In this graph, the x-axis showing age of flies and the y-axis showing the number of flies that cross the 10 cm mark. Also, the blue line showing the locomotor ability of Oregon R plus and the orange line showing locomotor ability of Park 1 mutant. You can also observe here that the locomotor ability of Park 1 mutant declined progressively with age of fly. Some of the applications of negative geotaxis assay are assessment of locomotor ability in Drosophila, to evaluate age-related decline in motor function and also to study neurodegenerative disease models and muscular defects. This video is made by me, Akash, and my batchmate Shivanshu under the supervision of Professor S.C. Lakhotia and Dr. Richa Arya, Lab of Cytogenetics, Department of Zoology, Banaras Hindu University.